Good afternoon. My name is Dr. John Thomas. I'm an associate professor of medicine at the University of Texas Rio Grande Valley School of Medicine, Department of Human Genetics. This is the Center for Genomic Research, short course, session 3, Genetics and Viral Disease, a genetic basis of human disease. So just as we've done for the other short course sessions, we're going to briefly examine a genetic topic that deals with virology and look at the implications for some genetic defects and how they affect viruses and viral diseases within people. Ideally, by the end of the session, you should be able to have a basic understanding of what an inborn error of immunity is and how these conditions predispose individuals to having severe or chronic viral infections. We'll describe and discuss some of the clinical implications of these, as well as examining some of the prophylactic and treatment options that are available. So this discussion is going to be based on a very broad literature search, which primarily focused on in vivo studies, as well as studies looking at clinical trials with patients that's been supported by molecular experimental evidence using animals or some in vitro work, overall with the goal of trying to characterize the physiological response and the mechanisms that govern these conditions. So I try to really use the most current research that's available in the scientific literature to examine some of these concepts. Most of the concepts we're going to look at today are going to focus around inborn errors of immunity, or IEIs. And these are basically genetic mutations that seem to predispose certain people to being more prone to certain types of viral infections. Many of these mutations occur within the immune system, and particularly within the innate immune response. And so we want to kind of try to examine what the genetic basis is that predisposes some individuals to being much more susceptible to viral infections than others. In addition to looking at the genetic mechanism behind this, we also want to kind of examine what are some of the clinical implications, including developing treatment therapeutic options for helping these people to either prevent or reduce the clinical consequences of their dysregulated immune response. Hopefully, the idea behind this is that we can help improve the patient's burden in terms of reducing the disease, as well as increasing their quality of life. And as we'll see from some of the discussions in some of the upcoming slides, some of these diseases and the defective immune response can have a very serious outcome for the patient. So particularly from a social perspective, none of these are necessarily going to be fatal, but they do cause issues. And so we'd like to be able to try to address these if we can. So we're going to look at these inborn error of immunities really within the context of both the innate immune response and then the adaptive immune response. If you're not sure what those are, I'd like to refer you back to short course session 1, where we did a brief introduction into the immune system, and we talk about innate and adaptive immune responses. So if you're not sure what we're talking about when we say innate and adaptive immunity, please go back and refer to short course session 1, which covers some basics of the immune response. So again, just to kind of remind everybody, and again, we covered in short course session number 2, some of the basics of viral replication and how viruses replicate. But just to kind of remind everybody again, when we're talking about viruses, they're going to have a very specific transmission cycle and a mechanism of pathogenesis that occurs when they infect the cell. So again, viral infections can really be categorized into 3 main phases: attachment, penetration, and uncoating. In the attachment phase, we have the virus that's going to interact with the host cell on the receptors on the host cell surface. Once this virus is able to attach to the receptor, it's going to be brought and internalized inside of the cell. This may not be mediated through clathrin-mediated endocytosis. It could be another pathway, but essentially the virus is going to be brought inside of the cell. Generally, it fuses with some kind of a vesicle, which happens with many products that are internalized at the cell surface membrane. This is a kind of a standard approach in cellular biology. Once this vesicle fuses with an endosome, there's going to be a pH change, which will then lead to the uncoating. So we've had attachment, we've attached at the surface, we've now penetrated and gained access to the cytosol. Now we have the uncoating process where the viral genome is now going to be exposed. It will then be able to go through the replication process, whether it's RNA or DNA, We'll eventually see the production of viral proteins. These viral proteins are then going to start to lead to the production of new viruses. And eventually we will see the virus. In this example, this could be a virus that's a membrane virus, an enveloped virus. And so it's going to become mature. It's going to eventually be directed to a place on the host cell surface where viral proteins are present. And the virus will then bud out or escape through this area. And now we have a new virus that's able to go out and infect new cells. So the basic life cycle, the virus has got to attach, it's got to get inside, it's got to uncode its genome and make new viral proteins and new viruses with the goal of eventually being able to escape the host cell. Just to kind of remind everybody again, this is a rehash a little bit from the short course session 2 on viral replication. Viruses have numerous replication strategies that they can use to replicate their genome and produce viral proteins. And many times this is going to be decided by the viral genome type that's first used to infect the cell. Is it DNA or RNA? Is it a single-stranded molecule, one, or is it a double-stranded molecule? Is it a positive sense or negative sense molecule, referring to the genomic polarity? Is it DNA RNA? Is it RNA DNA? There are several different configurations. And again, you can walk yourself through the 7 classes, and we explain this in greater detail in short course session 2. So if you're missing some of that information here, I'd refer you back to short course session 2 for viral replication. When we look at viruses in general, and this is just meant to be a kind of a representative diagram, we're looking at, in this example, the genome from a flavivirus. The flaviviruses are viruses such as dengue virus, Zika virus, yellow fever virus. These are all single-strand, positive sense RNA viruses that are transmitted through the bite of a mosquito. If we look at this cartoon here, we can see that the virus has a very specific organization of its genome. These are proteins that we refer to as structural proteins. These are actually going to produce the proteins involved in maintaining the physical structure of the virus. These are the non-structural proteins. These are proteins that are going to help the virus replicate its genome, package its genome, and maybe do some other small things, but they're not directly involved in the actual physical construction and maintenance of the viral genome. So we have structural proteins, non-structural proteins. These things are going to go through a variety of processing phases within the host cell. They're going to be cleaved, they're going to be chopped up, and then they're going to be reassembled to be able to carry out specific functions within the host cell, again, with the goal of being to produce new viruses and new viral particles that can escape and get out. So I put this up primarily just to kind of give you, the viewer, an idea for how complex this process can be, where we're going to have multiple events, multiple cleavage events and processing events. As indicated up here on the cartoon, these can be either host cell proteases that are going to be cleaving. These could be viral proteases through protease activity that's contained within some of the non-structural proteins that are going to process and cleave these things. The end goal being the production of new viral particles that can then be assembled and it can then escape and go out and cause disease. And again, if you're missing some of the detail here, I'd refer you back to short course session 2, where I went through viral replication in a little bit more detail. So we have the virus getting inside of the host cell. We have the virus beginning to replicate. Well, obviously, from the host cell perspective, this is not something we want to happen. How does the host cell address this? Well, the immediate response is going to be an immune response. So the host cell has an immune response. It has a system inside of it. And again, a short course session 1 does a very good job of examining host cell immunity. So if you're not sure how the host cell immune system works, refer back to short course session 1. But under normal conditions, in a person that has a fully functional, intact immune system, we would see viral replication occurring, whether it's a DNA virus or it's RNA virus, or maybe it's a mix of both. We have these viruses coming in, they're attaching at the cell surface, they're penetrating, they're uncoding and getting inside, and now we have maybe our DNA or our RNA that's now going to, when it gets inside the host cell, one of the first things that's going to happen inside the host cell in a normal, healthy cell is there are immune sensing molecules within the host cell that can detect foreign RNA or foreign DNA. and these are referred to as toll-like receptors, or TLRs. Again, this is mentioned briefly in the immune discussion for short course session 1, but we have these toll-like receptors that are going to be able to sense this foreign molecule present. TLR3 senses RNA, TLR7 senses DNA. There's some other TLRs that can sense other formats of these molecules, but basically we have this immune sensing system in a very non-specific way. The TLR can't tell whether or not this is dengue virus RNA or it's yellow fever virus RNA or Zika virus RNA. It only knows that it's foreign viral RNA. And so TLR3 is going to become activated, and it's going to activate a cascade of molecules that are going to be produced, which are going to lead to the production of other molecules. And ideally, we will get the production of interferons, in this case, alpha and betas. These are very potent immune signaling molecules that can be used to help modulate the immune response against the viral infection. In this case, we have interferon alpha and beta that are going to stimulate a pathway we refer to as the JAK-STAT pathway, which will eventually lead to the production of other molecules that are all designed to shut down viral replication. So under normal conditions, even if the virus comes in and begins to initiate the infection, we should see sensing molecules that are tied to the immune system inside the cell that should begin the immune response. Again, this is going to be a very non-specific immune response because the TLR is not going to be able to distinguish between dengue virus RNA and Zika virus RNA. It only knows that there's viral RNA present, and because of that, it needs to activate the alarm system and start to produce molecules that can help the immune system fight off the infection. So under normal conditions, this is what we're going to see happen. And in fact, if we look at this from a little bit granular detail perspective, and we look at certain cell types of the immune system, particularly dendritic cells that are exposed to RNA viruses, what we see is that these dendritic cells start to produce a broad milieu array of cytokines. These are chemicals that are designed to help increase and enhance, hopefully, but basically control or modulate the immune response. We're going to see that RNA viruses, through stimulation of TLR3, are going to lead to the production of a very specific range of cytokines that we don't see in cells that are not stimulated through TLR3. So these are RNA viruses up here that are infected immature dendritic cells. These immature dendritic cells, through TLR3 signaling, produce a very specific cytokine response that we don't see in substances that do not activate TLR3. And this LPS was a control down here. So we see the production of these molecules up here, these cytokines, at pretty high levels in response to TLR3. So we know that the TLR3 response, at least to RNA viruses, can be very robust. And for many RNA viruses, this will be a response that will eventually lead to clearance. So in a normal, non-immunocompromised individual, the TLR system is going to be a quick, easy to activate system, which will help the virus, help reduce the numbers of virus, the viral load in the immune system, while the overall immune system works to clear the infection. This is in a normal, healthy, non-immunocompromised individual. Sometimes, though, we do see deficiencies and defects in the TLR3 pathway, and this refers to what I referred to briefly before as an inborn error of immunity. So in this image, what we're seeing up here is a patient that's been infected with herpes simplex virus. Herpes simplex type 1 generally causes cold sores that appear at the corners of mucous membranes. Primarily, we see these lesions occurring at the corners of the mouth and on occasion at the corners of the nares. Mucous membranes are obviously very close to both these areas, and there's a lot of neuronal penetration in these areas. If you think about how sensitive your face is, when you scratch at the corner of the side, there's a lot of sensation there. You've got a lot of neuronal activity. Herpes virus infects neuronal cells, and it often alternates between a latent period of infection and a lytic period of infection. And again, I'd refer you back to short course session 2 when we talked about types of viral infections if you're not sure what lytic and latent types of infections are. So in a normal person that's got with a normal TLR3 pathway that is infected with herpes, you would generally see the formation of cold sores just here on the mouth, and that's it. In this poor woman's case, however, she has a deficiency in her TLR3 pathway. And when scientists went to examine, researchers did some studies to examine what this deficiency was, what they saw was that she's got deficiencies in both autosomal recessive and autosomal dominant genes that are involved in the construction of the TLR3 molecule. And in her example, these deficiencies in the TLR3 pathway lead to deficiencies in signaling, which don't produce the nice interferon response that we see under normal conditions. So in normal conditions, TLR3 is stimulated, interferon is produced, and it's going to help resolve the infection. In this lady's example, though, her TLR3 pathway deficiency is not letting interferon be produced. And because we're not seeing interferon produced or dramatic reductions in the interferon response, this has allowed this pathology to spread across most of her face. And as you can imagine, just from looking at these pustules are all more than likely containing virus. They actually, when they went and looked at the paper, and the paper reference is cited down here and it's included at the end of the talk, and swabbed these areas, they were able to recover infectious virus from many of these open pustules on her face. And you can imagine if you've ever had a cut or a sore on your face, how painful this must be, not to mention the social stigma that it would carry with it from having to go out in public. I'm sure this lady is very embarrassed at times when she has this to go out, which may lead to her not going and getting medication when she needs it, not doing other things she needs to do, maybe feeding herself, which could further exacerbate the condition. So just this small deficiency, this small genetic defect in the TLR3 pathway takes a herpes virus infection that would normally only induce a pathology right here in this very corner of her mouth and lets it spread across really the totality of her face, from her chin all the way up to her eyes. So in this example, when the TLR3 pathway is deficient, what we see in this inborn error of immunity is that because the TLR3 signaling pathway is deficient, we don't get the production of interferon gamma that we're looking for, and eventually we're going to have a much more robust RNA, a much more robust viral infection than we normally would. And because of this, we're going to start to see then virus being at levels that we normally wouldn't see. And in her example, and the example in the cases of others that have these inborn error of immunities, these genetic deficiencies, these deficiencies in the signaling pathway are going to lead to problems in regulating the viral infection. So this paper really confirmed that interferon is, which we've known for a long time, that interferon is a very important molecule in the host immune response against viruses, but particularly against herpes viruses, particularly those viruses that infect the central nervous system, which herpes is one of the classical cases for. So if we see that we have deficiencies in the TLR3 interferon-mediated signaling pathway, then this is really going to hurt the patient's ability to be able to clear or reduce the amount of the infection. So what we've determined is, what's been determined now is that this probably only accounts for maybe 10 to 15% of these cases. But if you're looking at this as a totality of people that are infected with herpes simplex virus, 10% or 15% of all the cases worldwide, this could be quite a high number. And so now what this has now stimulated is research into trying to understand what can we do to address these interferon defects, and are there any non-interferon associated pathways that could be stimulated to help shut this down? And so this is an area that's receiving a lot of intense research right now. And they're actually looking at other individuals, including several children that they found that have got these same genetic mutations, to try to see if there's something that can be done further up the chain on another pathway, which would maybe help to shut this down. So TLR3 deficiencies are significant error, and this is elicits significant error, inborn error of immunity in people that have these deficiencies in interferon gamma signaling. So in addition to having deficiencies in the TLR3 pathway, which leads to alterations in interferon production, particularly alpha, beta, but probably gamma as well, we also see genetic deficiencies in processes which are normal processes such as autophagy that can lead to amplifications of viral infection and kind of allow the viral infection to work well outside of the box, if you would, that normally would. So normally in the body, we have a process called referred to as autophagy, which is essentially the controlled breakdown and destruction of cells in response to cellular stress. So if the organism is going through a period of cellular stress, such as infection, inflammation, maybe times are tough and that you're not eating enough, so there's maybe some nutrient starvation, maybe oxidative stress if you're doing an intense amount of exercise, or you've got some other stress going on where cortisol levels are high, which can lead to oxidative stress. We have a process in the body called autophagy, which basically functions to help keep cells alive. And this is a response to basic types of stress. Normally, the autophagy process is going to involve sequestration. So we see the area that's receiving stress sequestered away from the rest of the body. Maybe these cells here are going through a lot of oxidative stress, so they're going to be kind of sequestered away from the other cells in the body. An autophagosome, the actual structure is going to be formed around in this cell that's going through this stress around the organelles and the cytoplasm. It's going to then be fused with a lysosome, which is going to involve this being degraded and broken down. And then all of these components that were broken down are going to then be recycled to then be used for other cells. Maybe the proteins are broken down to amino acids. The carbohydrates are going to be broken down into simple sugars. And all these things can then be reused. So autophagy in many respects can be looked at as kind of like the recycling process for the cells, for the body. Cell gets hurt, rather than destroy the entire cell and lose everything, let's recycle it and use those products to make new cells. So under normal circumstances, this process of recycling is going to occur, all over and over with no issues. However, in the context of infection, particularly viral infection, the virus may be able to use this process to help spread through the body. And in some individuals, just as we saw in the TLR3 deficiencies, we also see individuals that are genetically deficient in certain phases of the autophagy process, and viruses are able to take advantage of these breakdowns in the autophagy pathway in these individuals and spread and cause much more severe infection than normally would. So in 2020, a study was published that looked at this autophagy, this recycling process in humans that were infected with herpes simplex type 2, HSV type 2. Herpes simplex type 1 generally produces the pathology that we associate with cold sores around the mouth. Herpes type 2 produces a pathology that we generally associate with sexual transmission. So these are going to be the lesions that you would see in either vaginal or penile tissues in females or males. And this is the HSV that we normally associate with sexual transmission. So what we've seen, what researchers have seen is that in people that are infected with HSV2, and again, when you're infected with herpes, it's going to be more oftentimes than not, it's going to be a lifelong infection. This virus is going to fluctuate between lytic and latent periods of replication. There will be periods in your life where you couldn't tell you had herpes. You have no visible lesions. You don't feel anything. And then for whatever reason, the virus leaves that latent period. We think it's usually due to some kind of stress on the organism. Maybe it's oxidative stress. Maybe it's cellular stress. Some kind of stress event, environmental stress seems to reactivate the virus. It leaves its latent period and becomes lytic again. And when herpes replicates lytically, in the case of herpes type 1, you're going to see the formation of cold sores at the edges of the mouth, perhaps maybe around the bottoms of the nose and the nares. In the case of herpes simplex virus type 2, HSV2, this is the herpes that we associate with genital lesions. So you might see pustules and lesions forming on the vaginal or the areas around the penis and the groin. So in these individuals, the pathogenesis of this has really been kind of sketchy because in some people, they seem like they never leave the latent phase or never leave the lytic phase where the virus particularly stays active. And it's actually a very rare medical condition called molar A meningitis, lymphocytic molar A meningitis. And it's primarily caused by HSV2. And it seems like the virus is just consistently reactivated from the neurons over and over and over and never goes back into that latent phase. So whole genome sequencing was done on these patients, and they identified some very rare genetic mutations in some of the genes that are responsible for this autophagy, this cellular recycling event. And those genes are listed here, ATG4A and MAPILC3B2. Patients that have variations in these genes have deficiencies in the autophagy process. And because of these deficiencies, this reduces the amount of viral clearance that autophagy can contribute to, which leads to increases in viral replication and enhanced cell death. And they were able to restore these people to a normal function by supplementing in these normal copies of these genes. So they found people that had HIV or had HSV2 and they had deficiencies in these two genes. They were able to go in and restore the function of these genes temporarily, and they saw them able to have normal autophagy, normal production of these proteins, and the virus was cleared and shut down. So at least in these limited studies here, this shows that herpes simplex virus 2 is also using an altered autophagy pathway, an altered recycling pathway to be able to contribute to viral infection. So not only are we seeing deficiencies in TLR3, which leads to massive amplifications of viral infections, but we're also seeing this occurring in autophagy. And both of these are tied back to genetic changes in the host. So there are certain genetic deficiencies that are out there. There are certain that some individuals are carrying, which is going to make them more susceptible to certain types of viral infections. And not only certain types of viral infections, but this may lead to very exacerbated viral infections. If we refer back to the image that we saw of the young woman with the lesion over most of her face, that lesion should only really be on her mouth. So just a small genetic change can lead to a massive shift in what the clinical outcome is going to be for patients that are infected with these viruses that have these genetic defects. So a third type of inborn error of immunity that's been examined have been individuals that have mutations that are tied to the replication of DNA within the host. So just briefly touching on molecular biology, we've talked about the dogma of biology, which is DNA makes RNA makes protein. And there are certain molecules that are involved in this process. DNA has to get converted into RNA. There are going to be certain enzymes, DNA and RNA polymerases that are going to contribute to helping this process occur. And then once the RNA has been converted, or once the RNA has been produced, it needs to be used to produce proteins. So we have different types of molecules coming in that are participating in this. And one of the important molecules in this process is polymerase 3, is RNA polymerase 3. And within the context of varicella zoster virus, this is the virus that causes chickenpox in children and can cause shingles in older adults if you've already been infected with chickenpox as a child. And what they saw was that in some individuals, they seem to have a much more severe response to the infection from varicella zoster virus than other individuals. And when they went and looked at their genome and sequenced their DNA to figure out what was happening, what they saw was that in some individuals that had these, I shouldn't say some, in individuals that had these much more severe responses to VZV infections, what they found was that they had mutations in RNA polymerase 3. And as indicated by these red boxes here, you can see where RNA polymerase 3 plays some very important roles during the response to the viral replication cycle when it's inside the host cell. So In the case of the DNA virus coming in, which varicella zoster is a DNA virus, DNA viruses are generally going to contain areas that are very high in AT, in adenine and thymosin. We call these AT-rich regions of DNA. And normally, RNA polymerase 3 contains a sensor, if you will. The host cell can sense and detect these AT-rich stretches, and this is generally a signal that the host cell uses to say, hey, this might be viral DNA. Let's not replicate it. So just as we saw with the TLR3s, there is an innate, non-specific response to the presence of viral DNA, which is used to stimulate a host response to hopefully start to shut down and limit the infection. But in some individuals, this response of RNA polymerase 3 is deficient, and this AT-rich signaling event never occurs. And because we don't see the cytosolic sensing of the viral DNA occurring, this leads to a dramatically different type of viral infection and a dramatically different outcome for the host compared to what we would normally see in an individual that does not have deficiencies in RNA polymerase 3. And when genome sequencing was done to try to identify specifically what was happening, what was found is that RNA polymerase 3 serves several housekeeping functions in the process of making proteins, in the process of converting RNA into proteins. It's used to help transcribe ribosomal RNA and transfer RNAs. Again, these are RNA species that are involved in making proteins. That's beyond the scope of this lecture to discuss this, but if you need to, you can go back and look at some of the scientific literature or some of the information online. But essentially, we have RNA molecules that are used to help produce proteins, but they also serve a secondary role, which is they are there to be sensors for these AT-rich DNA fragments coming in, which may be from a virus. And when this AT-rich DNA segment is detected, there's a signaling pathway that's activated to produce interferon. Starting to see a bit of a pattern here. We got reduced interferon production through the TLR3 deficiencies. We got reduced interferon production through the autophagy pathway deficiencies. And now we're seeing reduced interferon production through the deficiencies in RNA polymerase 3 and its ability to sense these AT-rich viral DNA segments that are coming in. So because we're having this deficiency in RNA polymerase 3, this means that we don't have the detection of these AT-rich viral DNA segments, which means that we get a insufficient response. The immune system can't really respond and produce the proper molecules, and this leads to a dramatic increase in the amount of viral replication that's occurring. So again, we're seeing deficiencies in the immune system's ability to detect these molecules and then produce interferon to help shut down the viral infection. And because of these deficiencies that are occurring upstream, we're not getting the immune response that we need that's going to help shut down or limit the infection. And because of that, the infection begins to go out of control. And so some additional studies that were done using some female twins that were identified that both had this same genetic defect, and they both were diagnosed as being consistently, concurrently infected with recurrent. They were continually, constantly being reinfected with varicella zoster virus. And again, this was tied back to these deficiencies that they had in the inability of their RNA polymerase 3 to be able to activate the AT-rich cytosolic signaling, which would lead to the production of interferon gamma. Some additional studies that have been done that have followed up in other individuals that maintain this same genetic deficiency in RNA polymerase 3, and they've been able to further kind of identify some of these things, some of the mechanisms that are occurring. And there's still some questions to answer, but again, this is another example of where we're seeing the immune system from a non-specific perspective unable to generate a sufficient response against a viral pathogen because the host themselves has some kind of genetic deficiency. They have some kind of inborn error of immunity that's going to affect their ability to clear these viruses. So we've kind of talked about some effects on the non-specific immune response. But what about the specific immune response? Again, if you go back and look at session 1 for the immune system, we really talk about the immune system having 2 main phases. A phase that's a very non-specific response that provides protection, and a phase that's a very specific response that also provides protection. Again, non-specific protection is going to be a nice intact skin barrier. A specific response is going to be a T cell that can target a specific type of virus or a specific type of virally infected cell. So we've talked about these non-specific responses. Let's talk about some specific responses. So one of the specific responses that's been identified have been deficiencies in natural killer cells. Natural killer cells, and again, I refer you back to session 1 of the short course where we talk about this in a little bit more detail. But we have certain types of cells in the immune system, natural killer cells, B cells, and T cells, that are all designed to kind of go after specific pathogens based upon the pathogen that's replicating inside of the host at that time. And what's been shown is that, and the the example that was used in the literature was Epstein-Barr virus. Epstein-Barr virus is a double-stranded DNA virus that can lead to the production of a form of leukemia that can be very severe, as well as some other diseases. And Epstein-Barr virus is one of these viruses that alternates between a lytic and a latent cycle of replication. Again, we're starting to see a bit of a pattern here. Herpes virus, lytic and latent. Varicella zoster virus, lytic and latent cycles of replication. Epstein-Barr virus, lytic and latent cycles of replication. And it is when the virus starts to replicate in its lytic cycle that deficiencies in the immune system of the host can start to pop up. And so in Epstein-Barr virus, what they've seen is that many of these inborn errors of immunity are related to how T cells and B cells interact. And what we see is actually a disruption in some of the molecules that these B cells and T cells use to signal and communicate with each other. So if they can't signal, then they can't help shut down the infection. And this results in a very severe form of the disease that's often going to be widespread throughout the host. One of the purposes of the immune system when an infection occurs is to try to limit the spread of the infection to a region within the host by recruiting all the immune system cells that are available to that area so they can shut down the infection at that spot, not allow the infection to spread and disseminate throughout the body. But if you have deficiencies in B cells and T cells because you have some kind of genetic deficiency and you're infected with Epstein-Barr virus, you're not going to be able to limit the spread of the virus. And so it replicates everywhere. And eventually, this can lead to the production of lymphoma. And this can lead to also the production of severe types of immunodeficiency diseases. In individuals that are infected with cytomegalovirus, CMV, again, another virus that is infected in most individuals are infected with, that latently persists throughout the life cycle of the individual. But for some odd reason, these B and C T cell signaling deficiencies lead to CMV producing a severe immunodeficiency disease and starts to madly proliferate and disseminate throughout the host. And now that they've identified this mechanism, they've been able to determine that other viruses, including certain types of herpes viruses, Kaposi sarcoma, which is a virus that we normally see in people that are infected with HIV, also have deficiencies. We see other deficiencies that lead to reductions in the production of interferon, reductions in the activation of other pathways, and all of these, again, lead to a very severe dissemination of the virus and a disease. So these inborn errors of immunity seem to particularly impact the ability of the immune system to sense and respond to these pathogens. And when this occurs, you get essentially what's could be described or viewed as unchecked viral replication throughout the host. If we look at these adaptive immune deficiencies, we can see, again, as indicated up here on the slide, these are all coded in red. When you have a T cell that's maintaining some of these deficiencies, these genetic deficiencies, some of these inborn areas of immunity where the T cell can't function and signal properly, you lead to T cell anergy. This is basically the T cell hanging out in an inactive state, not able to go out there and perform its function. The same thing can happen with B cells. We need B cells to be able to produce antibodies, but if these B cells cannot receive signaling from other cells of the immune system, particularly from T cells, because they may be deficient in some of these receptor interactions, then you're going to have B cells that can't produce antibodies. You could also have these T cell deficiencies that come back into the immune system signaling pathway and downregulate dendritic cells. So if we have this pathway where we have dendritic cells stimulating T cells, stimulating B cells, with the goal of eliciting a sterilizing or a pathogen clearing immune response, but there are inborn errors of immunity, genetic deficiencies in the ability of these T cells and B cells to signal and communicate with each other, then we're not going to get clearance of the pathogen. We also do see this to some extent with people that sometimes take live viral vaccines. I won't spend too much time on this because by and large, we don't use live virus vaccines in humans. We do use these a lot in animals, but we don't use these a lot in humans. Now, is this a 100%? No. There are some live virus vaccines that are deployed, but they're very, very rarely used, again, primarily because of the liability issues that would come along with it. No pharmaceutical company wants to get out there and put out a live virus vaccine that has the ability to cause and replicate within an individual. So, but we have, in a few times that live virus vaccines have been deployed, we have seen some individuals reacting to them. And this does seem to be a very narrow segment of the population. But again, when we've looked at these people genetically and sequenced their DNA, what we see is that these deficiencies are tied to deficiencies in interferon production. So some genetic deficiency leads to a downregulation of the production of the interferon from the interferon production pathway, and this allows the virus to be able to spread and replicate pretty much out of control. And this has been seen for several different types of viruses: measles, mumps, rubella. We've seen this in people that have received yellow fever vaccines, that received the MMR vaccine. So we do see that this occurs in a small segment of the population. And again, when they've gone and looked at these people, they see they've got some kind of genetic deficiency in the JAK-STAT pathway, some kind of deficiency in the pathway that leads to the production of interferon gamma, which just kind of, again, emphasizes how important the role of interferon is, alpha, beta, and gamma, both types 1, 2, and 3, in controlling viral infections. So we've talked a lot about the bad side of these genetic deficiencies, right? You've got a genetic mutation, and at least from the context of the discussion so far, these mutations have all been bad. Is there ever a condition or a scenario where a genetic mutation could be beneficial? And the answer is yes. And so we're going to end the talk here with looking at a couple of these benefits that the host can receive through a genetic mutation. And I think probably one of the most famous ones is referred to as Delta 32, which is what we'll spend the rest of the time talking about here. So we have identified some individuals in the global population that contain genetic mutations within their immune system that actually gives them protection or some benefit within the context of infections, and particularly within the context of being susceptible to infection with HIV. So there was a study that was done in the late 80s, early 90s, looking at a segment of people within Thailand. Most of these people were sex workers. At the time, Thailand had a very, very high seroprevalence for HIV. I remember in the late 80s in Thailand, in certain areas of Bangkok, 3 out of 4 people were infected with HIV. There was a massive, massive level of HIV infection at the time. And because of this, Thailand received a lot of focus and a lot of scientific study on HIV. And a population of sex workers was identified in Thailand that seemed to be resistant to HIV. They had had known contact with individuals that had HIV, sexual contact, and yet these individuals were not infected with HIV. So obviously the question was, why? why are these people resistant to HIV? And what they found was as they read and looked at their DNA, they found that these people had a small mutation in one of the receptors, CCR5, that's a receptor that's used by the HIV virus to gain access to the cell. That's part of the attachment and the penetration phase is binding of HIV to CCR5. But in individuals that were resistant to HIV infection, what they found was that they had about a 32 base pair deletion in their CCR5 molecule. And because that gene, that CCR5 gene deletion was there, it gave these people resistance to HIV. And we think that this resistance primarily came because the size of the CCR5 molecule, as indicated on the cartoon, was altered. And in an individual that has a normal CCR5 gene, the protein was big enough to allow HIV to stabilize along with CD4 and gain access to this cell. In individuals that have this delta 32 mutation, delta meaning different or deleted in this case, 32 meaning the base pair, in individuals that have the CCR5 delta 32 mutation, they don't produce CCR5 that's functionally able to interact with the HIV virion. And so these individuals cannot get infected with HIV. They can have HIV virus floating around in their bloodstream, but the virus is unable to gain access to a host cell. And as we all know from our discussions on viral replication in short course sessions 2, without the ability for the virus to gain access to the internal mechanisms of the cell, the virus is a parasitic organism. If it can't use the host cell replication machinery to replicate, then the virus is not going to be able to replicate, and that therefore we won't have a productive infection. So this mutation has gotten a lot of studies since it was identified about 30 years ago. People have used it to try to generate inhibitors. And we'll actually talk about some of the good stuff that's come from discovering this delta 32 mutation. So as I mentioned previously, when delta 32 was identified, this really helped give some impetus to pharmaceutical companies in the sense of looking at ways to block HIV binding. So I wanted to kind of close with this as kind of being a good case scenario for an inborn error of immunity, a genetic deficiency in the immune system, actually giving the host a bit of a benefit and actually kind of indirectly helping all of us benefit from the identification of this mutation. So when the delta 32 mutation was identified, what this led to was the production of now inhibitors and other molecules that can be used to block or interfere with different phases of the HIV replication cycle to help the host be able to survive the HIV infection. We all know that HIV eventually will progress into AIDS, acquired immunodeficiency syndrome. And once someone has AIDS, their immune system is going to completely shut down. And this is where you're going to be susceptible to any kind of infection at all. And usually this is going to lead to a dramatic reduction in the lifespan of the host. But people that are infected with HIV, we put them on a cocktail of different drugs that we refer to as heart therapy. And these drugs all interfere with HIV at different phases of its replication cycle. One of these drugs is a drug types is referred to as a chemokine receptor antagonist, and it's used to block HIV fusion to cells at that CCR5 molecule. So this is an example of where we were able to kind of use the identification of this inborn error of immunity, this delta 32 mutation, to benefit us in terms of drug discovery. And so there is something that can be beneficial from identifying these inborn errors of immunity. And this does allow us to be able to generate not only a new class of drugs that can maybe help extend the lifespan with this person, but it also expands our understanding of the life cycle of the virus in general, which may allow for further opportunity, as we've taken with HIV, to interfere at other segments of the life cycle to be able to allow the host to survive more. So we've got chemokine receptor agonists that can block CCR5. We've also got protease inhibitors that can help block the formation of the virus itself. We've got integrase inhibitors. HIV is a retrovirus, which means it has to integrate some of its genome into the host cell. If you can block that process, you will stop the infection. Now, the good part is that we have a nice wide array of drug types that can interfere at different phases of the replication cycle. The bad news is that because we have these drugs that are used, eventually the virus does adapt to these different drugs. And so one of the concepts behind heart therapy is that we give a cocktail of drugs and we rotate them. So we give you maybe these 2 or 3 drugs for through 2 or 3 months, and then we'll pull you off them and then give you these 3 for 2 or 3 months. And then maybe we'll go back in 2 or 3 months and then give you these 3 or these 4. We try to rotate and vary the different drug types that are being used to limit the virus's ability to adapt, because the virus will eventually adapt. And we do see this even in heart therapy. Eventually, the HIV virus does adapt to these drugs and they largely become ineffective. But what we have found is that by varying the drug types and by changing up the therapy, we can take the utility of these drugs, which initially was only maybe a few years, but now because of our understanding of how the virus replicates, and because of our understanding of how some of the inborn errors of immunity and these genetic deficiencies function, we've now been able to take this heart therapy and take it from only being effective for maybe a couple of years to where it can now be effective for 2 or 3 decades. So there are some benefits that we have gained from examining these inborn errors of immunity. And this is probably the approach that we will continue to lean on as we identify more of these. There may be many viruses and many viral infections that we just don't know at this point that are being supported by certain errors within the host immune system that are at the genetic level that we're just not aware of. And hopefully these studies will continue to stimulate discussion and research into these mechanisms. And this completes short course session 3.